and I recognize this is a pretty diverse audience. Um, so what I would ask is that you stop me if we don't get through everything. That's totally fine. Um, I uh, but I want to make sure people understand what I present, and so I will work my way through. Um, and thank you so much for having me on to speak. Um, and I want to start especially by acknowledging two of the people who really a lot of the content I'm going to present on the talk really comes from these two individuals. The particular paper you asked me to come and speak about was about a method called GeNA that was largely developed by Lori Rumker. She's the lead author on that paper. She's a former MD PhD student who has now finished her PhD and is now still engaged with the lab as a sort of part-time postdoctoral fellow as she transitions into clinical work. And Yakir Reshef, who's formerly a postdoctoral fellow, but now is a research fellow in medicine. He's a rheumatology fellow. So a lot of what I'll be talking about is based on the framework that Lori and Yakir developed together. Um, uh, and as a lab, we focus a lot on autoimmune diseases. That's a major component of what we do. These are diseases where the immune system attacks different organs. Some of these diseases can be very organ specific. Multiple sclerosis, for example, is a disease where the immune system attacks the brain. The particular condition that we spend most of our time thinking about in our group is rheumatoid arthritis. It's a disease where the immune system attacks a very specific tissue in the joint called the synovium. And you can see this thin membrane in the healthy joint becomes engorged and red on the right. So that's what happens when the immune system interacts with the synovium. It gets thickened and then slowly destroys the joint. It's a disease that affects up to 1% of the population throughout the world and leads to the destruction of joints of the hands and other joints as well. And it's mediated by immune infiltration of the synovium. It's characterized by autoantibodies and can have systemic complications. Um, so our group takes multiple strategies to trying to understand the mechanisms of human diseases. One thing we like to do is to actually look at the tissue and look at the inflammatory architecture of the tissue. This typically involves us wanting to define the key pathogenic cell states in disease. Another thing we do is we try at the same time to think about human genetic mechanisms. So the genetics are the differences in genetics that we all have are what we're born with. So those risk alleles are unequivocally causal. They define key disease mechanisms. Um, the downside of human genetics is while they point to causal mechanisms, defining the function of those genetic differences between individuals that puts one person at greater risk and one person at lower risk of a disease. Finding those alleles with genome-wide association studies has gotten easier, but understanding what those alleles do continues to be difficult. And both of these approaches require careful and rigorous computation and statistics. A lot of the work I'll be talking about here today and the methods that I'll be discussing are really very related to the explosion in single-cell genomics. And as you can see, since about 2010 to today, we've gone from assaying a small number of cells to assaying thousands and thousands and thousands of cells in a single assay. And very recently, since about 2017, 2018, the 10x platform has been the workhorse for this field. It allows encapsulation of cells in oil droplets, and those oil droplets also have the sequencing reagents. So this is what's really made it possible to assay 5,000, 7,000 cells per experiment, and has really allowed us to query not just a small number of tissue samples, but actually a massive number of cells from many samples, indeed from whole cohorts, which enables us to identify cell populations that are different between affected individuals and unaffected individuals. It also allows us to identify the effects of genetics, right? Those alleles that put you at risk of an autoimmune disease. We're able to use the single cell data to say what does that allele do at the level of gene expression, also at the level of cell populations. And when we think about single cell data, any of you who've worked with this data will know that the data is complicated. It's often a victim of batch effects. The data can be very sparse. In many instances, when we collect data from many individuals, we need to make sure that the data across the different individuals are comparable to each other. And then we also need to have a framework to find the differences in single cell phenotypes, both across disease subtypes and between different genotypes. So the two approaches that we sort of take is one, looking at the tissue, as I mentioned, identifying key cell states with single cell genomics in rheumatoid arthritis, and trying to understand what the architecture of inflammation is. And then the second is looking at genetics. And I'll just summarize some of the high level work that we've done in RA before I dive into some of the methodological challenges. So let me start by talking about tissue. So as I mentioned in rheumatoid arthritis, this is a joint, and you can see the most inflamed area here is the synovial tissue, which is filled with immune cells, changed tissue cells, fibroblasts. So we want to know what's going on. And one of the key questions that we're interested in is all RA the same, or are there differences? And we know already before we did this study that there's some key populations that are important in RA. For example, granzyme K T cells, CD4 T peripheral helper cells, the so-called autoimmune associated B cells, inflammatory fibroblasts. And the question is there some kind of structure, right? Are all these pathogenic cell states increased in all patients, or are there subgroups of patients that have some enrichment for these different groups? And we want to know if we can sort of deconstruct disease and explore tissue heterogeneity in rheumatoid arthritis. So working with the AMP-RA-SLE consortium, we assayed 82 synovial tissue samples and obtained data on 328,000 cells. We
Okay.
I think that's a noise, like it wasn't intended to be a question.
Oh, okay. I'll keep going then, unless
Yeah.
someone has a question. Please do stop. So we looked at 328,000 cells. As you can see, we have natural killer cells, T cells, stromal cells, endothelial cells, so all of the major cell types. And indeed, what we could see right away is that there were differences in our patients. And we call these cell type abundance phenotypes. It's a very straightforward thing, really. We saw that there were some patients who had really mostly myeloid cells, some patients that had a mixture of T cells and myeloid cells, some patients that had a mixture of T cells, B cells, and myeloid cells, and other patients that really had many fibroblasts. So, and in this way, we found 6 different groups. And naturally, we would want to know what the differences are. And then obviously, we could take, say, the T cells themselves, as we've done here, and we could cluster these cells based on their RNA-seq profiles. And you can see that we found 24 different T cell clusters. And then we could go through and try to understand what those populations are by looking at what genes they express, what protein markers they express. And here, for example, we've labeled a cluster, cluster 8, as regulatory T cells because of the fact that they're CD4 expressing, that they're CD25 positive, which is a marker of T cells. Okay, so And in this way, we could look at each of these different cell types, T cells, NK cells, etc., and subcluster them all into these groups of different cells. We would call them cell states. Um, and try to understand how what sort of a role they're playing in RA. So the natural question we want to ask is, um, we have all these different RA phenotypes, these CTAPs. So for example, there's some people who have lots of T cells and fibroblasts, some people that have lots of T cells and B cells, some people that have lots of T cells and myeloid cells. A natural question to ask is whether the types of T cells in each of these groups are the same or if they're different. Um, so for example, T peripheral helper cells or regulatory T cells, are they increased uniformly across these different groups, or are there some groups that have more regulatory T cells? And it's important to know biologically, because if that's the case, it would influence perhaps how we think about treating these different diseases by going after different cell states. So that's what's going on the tissue side. On the genetic side, we think it's really important to think about genetics and finding risk alleles. Can genetics help us identify new therapeutic targets? Can we define drugs that go after particular genes? Perhaps if we know that there's a genetic risk factor that's useful. There's already strong evidence that risk genes, based on genetics, are putative drug targets. They're more likely to work out in pharmaceutical development than not. Um, and we see in the last section, I talked about how we were hoping to find different cell states, but it's hard to know if a cell state that's expanded in disease or in a particular disease subtype is really driving the disease, or if it's sort of reactive, if it's a passenger, if it reflects the sort of damage that's happened from the disease. And so genetics points to these kind of causal mechanisms and helps us understand what's driving inflammation and what isn't. Um, so we think that these risk alleles and risk genes may be acting in key cell states and that they may be expressed in key cell states. And so we've been involved in genetics over the year. This is our latest study, which was done with myself and many collaborators around the world, but especially by Kazuyoshi Shigaki, Saori Sakawi, Chikashi Terao, and Yukinori Okada, who were my kind of key collaborators. And as you can see from this review, we now have over 100 risk alleles associated with rheumatoid arthritis, many, many, many different genes. But really, in most instances, what we're interested in is understanding how those genetic changes would alter a single cell phenotype. And of course, in most cases, we don't know that. And part of why we developed the method, the GeNA method, is to try to answer that question. So in terms of single cell methods, there's a couple issues that we've been really thinking about. The first is that data sets, as we obtain them, need to be comparable. So we need good data integration methods to make sure that our single cell data from different individuals can really be compared to each other, can be looked at all together. And then we need to be able to have a framework to find differences in single cell phenotypes. If we have such a framework, we can ask, do we see differences between different disease subtypes, between cases and controls? And we can also ask, are there differences if we stratify by genotype? So if you take somebody who has a risk allele versus not, do we see those differences in the single cell states as well? Let me start by talking a little bit about making data sets comparable. And this is work that we've done over the years with Ilya Korsunsky and others in the lab. This work started when we started looking at synovial tissue fibroblast data sets. And you can apply kind of standard single cell analysis strategies. And what you can do is you can cluster your major cell types, fibroblasts, endothelial cells, parasites. And that looked very encouraging. But now if you started coloring your cells by the individual you got them from, you start to see that every individual sort of clusters out separately in this UMAP plot. So cells from an individual were more similar to themselves than cells from another individual. And what we thought that really reflected was not inter-individual differences. We don't necessarily believe that every person has their own unique cell states, but we think that reflects more kind of batch effects, that each of these samples were handled slightly differently, processed slightly differently, and that created some difficulties. This is another paper around the same era, looking at some lung tissue. Again, you see these major clusters, but if you start coloring by individuals, you start to see that each of the individuals sort of segregate. And again, that's a problem. So this is early data from the Human Cell Atlas, looking at T cell heterogeneity. Again, you apply standard methods. You see that there's a bunch of different clusters of CD3 T cells. There's some of them are CD8 positive, but it's color by individual. You see that the individual T cells from each individual person is kind of clustering out separately, which again is not ideal. So why is this a problem? So we think that there are confounders. We think that there are technical confounders that affect different cells differently. We think that data sets have varied cell type compositions. And as we try to correct away batch effects for standard data, you might use some kind of linear regression and regress away all your problems. But here, I think the problem that we have is we think that the different cell states or cell types are affected by batch and technical issues differently. So T cells may be influenced differently than, say, B cells. So Ilya Korsunsky developed a method called Harmony, which we published in 2019, to address this method. And what we start by doing is we take all our single cell data and we apply soft clustering. So we get a sort of soft assignment, and every cell can be assigned to really to multiple clusters. Then for each cluster, we get a centroid, and we correct the centroid. And remember, every cell has a complex identity because of soft clustering. It belongs to multiple soft clusters. So every cell has its own degree of correction. And then on the right, we go ahead and then make a correction of each of the soft clusters across the technical batch effects. So this method is a very powerful, very fast method. It's become one of the most popular batch correction methods for single cell data, and it's been very useful to us because we've had to correct away all these kinds of problems in our data. Here's a simple example. We're looking at GeRCat cells and 293T cells. These are two different cell lines. You can see we've got three experiments here, one that has only GeRCat cells, one that has 293T cells, and one that has a 50-50 mix. And what you see that happens when you use the 50-50 mix, the cells go cluster separately. And they're poorly, like the cells that are from the experiment with GeRCat and 293Ts, they cluster separately from the GeRCat cells that are just from the GeRCat. When we apply Harmony, you see that there's nice clean mixing where the GeRCat cells all mix together, 293T cells mix all together. This is that same data from the Human Cell Atlas. This is 500,000 cells. Cord blood and bone marrow. You can see that they're mixing together nicely. And now we can look at the individuals. You can see all the individuals mixed together really nicely too. And now we can go through and annotate the cells with known cell types and cell states. So just a quick summary about data integration. And again, this work is slightly older, but extremely important for any kind of single cell analysis. We propose an algorithm called Harmony, which is fast, scalable. It's a single cell integration method. It's able to correct away technical categorical variables and inter-individual differences, batch effects, and platform differences. And this effective integration can allow us to annotate cells. It's essential for novel biological discoveries. And it allows us to kind of cross-compare individuals to each other. So for all the analyses I'm going to show you, we've always used Harmony first and then built other analyses on top of it. Okay, so now we need a framework to find differences in single cell phenotypes. And a lot of the slides I'm presenting here and the content here is courtesy of Lori Rumker. And when I, and Yakir started working together, we had some analytical questions, which is how is disease tissue different from control tissue? How do we think of treatment? If we have data on patients before they're treated, can we tell what predicts who's going to respond, who doesn't respond? If we look at tissues before and after treatment or after disease progression, can we find the cell states and cell types that are really changing in the most dramatic way? So we want to be able to do these cross-sample comparisons. The benefits of these is that we can understand how diseases develop. We can characterize heterogeneity between patients with it, and hopefully we can So I think for a lot of things we've done in biology, we're used to this sort of framework where you've got a whole bunch of samples and you have a whole bunch of features that you measured. In the world of transcriptomics, the samples might be individual samples you've obtained from disease, and the features might be measures of individual genes from a tissue sample or proteins for that matter. Single cell data, we have a different situation. We have multiple samples here, and for every sample, we have many, many, many cells, and for each of the cells, we have many genes. So we actually have many profiles for every sample, and not necessarily the same number of profiles because we get different numbers of cells from each sample. So ultimately, when we do these analyses, we want to be able to take samples and look at different features. And the question is, what features should we be looking at? One very simple thing you can do is you can average each sample and create a pseudobulk. So you can take all the cells from sample A and just average them together, all the cells from sample B, average them together, all the cells from sample C. So needless to say, you lose a lot of information by doing that. But it does get you to the point where you have individual gene measurements for every sample, and that allows you to do sort of standard comparisons. The other thing you can do is you can cluster cells together. So here you can see Lori has indicated that you can cluster T cells together, B cells together, and so on and so on. And so for every sample, you can say, well, how many T cells do I have? How many B cells do I have, et cetera, et cetera. And that you might be able to do that and then make comparisons and say, okay, do my cases have more T cells, fewer B cells, et cetera. So now you're doing an analysis at the cluster level. The limitation of this approach is it's hard to know if the clustering approach you use matches relevant biology. And these clusters are constructed based on intercell rather than intersample variation. So you're grouping cells together just without taking into account how they vary across samples. And of course, once we have a matrix like of samples and features, we can do unsupervised analysis, we can do supervised analysis, or some combination of both. And again, the framework I'm going to present now is this framework that Lori and Yakir really developed and then applied to different situations. So let me start by talking about transcriptional neighborhoods and our sample features that we're going to use. And then in the latter part of this, I'm going to talk about how we do case control analysis and also specific analyses about genotype-based work. So for a second, I'm going to ask you to forget the fact that we have so many different genes and just imagine that for every cell, we have just 2 genes. And you've got now a bunch of samples, sample A, B, C, and D. And what you might see is that sample A and sample B look very similar. There's some cells that express both gene 1 and gene 2 in the upper right, some that express fewer gene 1 and gene 2, and the individuals that have lots of low gene 1, gene 2 expressors also have a lot of high gene 1, gene 2 expressors. For sample C and sample D, you have a slightly different situation. These individuals have cells that are low gene 1 expressors and high gene 2 expressors, and also low gene 2 expressors and high gene 1 expressors. So what you see is that there may be two sort of cell expression programs going on here. And the people who have a lot of these upper right cells also have a lower left cells. And the people who have the upper left cells also have a lot of the lower right cells. And of course, these are density plots. You can look at these individual cells. And now one of the things you can do is we can take this transcriptional space and cut it up and ask simply how many cells are in each of these spots. So here we have sample A. We've made this kind of simple grid. And actually what we can do is center a neighborhood around each cell. And then start asking what's around every cell, not just in sample A, but across all other samples. And we call these neighborhoods. So rather than the unit of analysis for a lot of what we do is not clusters, not these large clusters, which are of arbitrary size, but rather these sort of small neighborhoods. How do we define these neighborhoods? Well, what we do is we take our nearest neighbor graph. So for every cell, we can define which cells it's most similar to and build a nearest neighbor graph, which is very commonly done for single cell analysis. And then what we can do is start doing a random walk and seeing how many steps does it take for me to end up at a very specific cell. So one step might end you up, from a small number of cells, you might end up at our target cell with one step. With two steps, more cells get there, and with greater steps, more cells get there. So we get a probability density across the cells in the graph, which reflects the likelihood that you'll arrive at your anchor cell after k steps. So now what we can do is for every neighborhood, we can say how well represented is a particular sample in the neighborhood, because each of these cells are coming from different people in our very well integrated nearest neighbor graph. So when we define the neighborhood, as you can see, as you take more steps, our neighborhoods slowly get bigger. And each of these neighborhoods are occupying a different space in the transcriptional landscape. So, okay, so that's how you take cells and how you define neighborhoods, but how do you define sample features? So, as I've sort of alluded to, what we're doing now is taking a whole bunch of our samples, and for each of these neighborhoods, and remember in our data set, we have as many neighborhoods as we have cells, we can say how well represented is each sample in the neighborhood. So now we have a certain number of features. You might have 100 samples, 1,000 samples, but you may have 10,000 or 100,000 neighborhoods. So it's a short but wide matrix. And obviously, there's a lot of redundancy in the matrix because many of the neighborhoods are going to correlate together. They'll correlate because they're very close to each other, or they correlate just because when you have more of a certain cell state, you have less of another one, or you have more of a certain cell state, you have other cell states that go along with it. So we apply some kind of matrix decomposition as illustrated here. And our typical thing that we use is principal components analysis to capture the major variation in this matrix. So here you might imagine that your first principal component in this kind of example, before we were talking with individual genes, now maybe we'll talk about it in a more abstract space. Some of your samples may have this positive loading because we saw before that this axis had cells that went together. Others may have negative loadings because we found that other individuals, the individuals that had fewer of these cells, had more of these blue cells. We might find other PCs that are less important or equally important, but that capture more subtle differences in the transcriptional space. So these NAMPCs become really important. They become our kind of sample features because they reflect that neighborhood abundance for these hundreds of thousands of neighborhoods, but they aggregate effectively the neighborhoods together that are most similar with each other. And now what we can do is take every sample and think about the NAMPCs specifically and focus on that as our way of understanding the features. So how can we use this to do case control analysis? So we use a method that we call covariant neighborhood analysis, or CNA. And this allows us to really think about disease subtypes and comparing them with each other or any other sort of sample level variable. So here we imagine that we have data for every sample, for example, clinical status. For RA, we were talking about these different subtypes. So it might be, are you in a particular subtype or not? But you can also imagine looking at other demographic features. And now you remember we have those NAMPCs, which largely pick up on which neighborhoods in the cell transcriptional space, which cell states effectively, are captured by these different samples. And now what we can do is do a simple linear regression analysis. And that'll tell us if that turns out to be significant, if the cell states are different in a way that's informative for this particular phenotype Y, which again might represent a clinically important issue. Some of the topics I won't get into, but are well handled in the Nature Biotechnology paper, is how do you choose the right number of PCs? Obviously, in many instances, we do want to use covariates. We want to account for batch, for example. And in practice, what we do is we use empirical p-values via permutation rather than analytical p-values that you might get by linear regression. So we're able to calculate case control comparisons. And once we have them, and once we know which PCs matter, we're able to then figure out which neighborhoods are driving the signal. So we get results like this. So this is a study where we looked at TB, and you can see that there's some T cell states that are red. They're associated with people who are likely to have active TB. Some individuals are blue, or some cells are blue. Those are the cell states that are depleted in the individuals that are likely to have active TB in this case. Obviously, we did a lot of simulations to understand our method. This is one very simple example of a practical example that we looked at. These are individuals who've had sepsis, and we looked at data in cases of 29 individuals and controls. It's a paper from the Nir Hacohen group, and what they found is that this one cluster MS1, as you can see over on the right and indicated in the red in the lower right plot, is expanded in sepsis. Now, when we apply CNA, we see like a little more, right? So we see that indeed some cells are expanded with sepsis, other cells are depleted as people develop sepsis, and it doesn't line up perfectly with the unbiased clustering. In fact, what you can see in that MS1, our results largely agree that they thought MS1 was expanded. We saw that a lot of the MS1 cells, that cell state was indeed expanded, but we saw that MS4 also had some expanded cells, but some depleted cells. So there's heterogeneity. So MS4, that cluster is largely obscuring a lot of interesting things that are going on. And then there's some other clusters. And we're actually able to look at these individual neighborhoods and say which ones are expanded with sepsis, which ones aren't. And you can see we're getting at a much finer resolution here. Rheumatoid arthritis, I showed you a large single cell data set. What about those RA subphenotypes? Are they different in RA subphenotypes? Well, we can go and apply CNA. These are natural killer cells. This is the clustering I told you about. So we can take these clustering, each of these clusters, test them individually. Whoops. And I'm going to talk about this T plus F phenotype. And you see some clusters are expanded, some are not. These all have z-scores of 1 to -1, so they're not individually that statistically significant. Probably the clusters are too small because you see some of them are read together. You can see the major difference in this group are CD16 positive, CD16 negative. Now you apply CNA in the upper left, you see that there's a bunch of cells that are blue that are really depleted in this T plus F group, and a bunch of cells that are red, which are really expanded in this T plus F group. So in this case, the clustering that we did, the transcriptional clustering was too fine, and then CNA gets you again to the right answer, gets you to more coarse resolution, which is what turns out to be important. And again, this corresponds to CD16 expansion in the synovial tissue for individuals that are T plus F. And these are actually reflects interferon gamma biology. I won't get into that. But now we can go through and start looking at our different cell states and looking at our different phenotypes, and we can see for NK, some cell states are expanded in this particular phenotype of RA patients, others are depleted. But now we can also go fill out this table as we have in our 2023 Nature paper. That's how we think about case control, but what about genotype? Well, we can use the same framework. So how does an allele affect cell state? How would we answer that question? We would apply the same framework or a very similar framework to genotype. So now this is the GeNA method, thinking about genetic association with genotype neighborhood. Genotype neighborhood analysis. And again, this is work led by Lori, and Lori set up a collaboration with the 1K consortium, obtaining 969 sample profiles. We had something close to a million cells to look at, a million single cell profiles. And what we really want to ask is for an individual that had AA or TA or TT for a given SNP. So here you have one SNP, the 2 chromosomes. Does that affect cell state? Now we know from a lot of studies from our group and other groups that SNPs can affect the level of expression of an individual gene. If a SNP alters the expression of an individual gene, perhaps it can alter expression of a protein. But what does it do at the tissue level? Are there cell states that are affected? Are there selected cell types that become more abundant or not? Are there more subtle changes at the cell state level? And that's what we want to answer with GeNA. So GeNA identifies cell state abundant quantitative trait loci in single cell data. I won't present this in great detail, but it has a well-controlled type 1 error, and has a good statistical power. And here I'm going to talk about how we applied it to a real data set, this real data set of the 1K, and we tried to replicate our results in independent data sets. And in some cases, we found examples of alleles that colocalize with immune-mediated diseases. Here's the method. So just like before, we take advantage of those neighborhood abundance matrix PCs, and we have allele dosages, and we look for, we regress these things together, and we look and see if for a given allele, we have an expansion or depletion of a particular cell state. And that allows us to link SNPs to cell states. And here you can see we found in NK cells and myeloid cells a few examples of SNPs that seem to be altering the balance of cell states in individuals within our data set here. So we were very excited about this. Here too that you're seeing, you can see they're both in NK cells, but you can see the effect of these SNPs, even though the SNPs are very close to each other, they're they're on different chromosomes. They're subtly they're different, but similar. Here's one SNP that we spent some time thinking about, and you can see that if depending on which allele you have, if you have the TT allele versus the CC allele, you see an expansion of the red with the T. In more T alleles, you see an expansion of these red cell states in NK cells. And it's very interesting because when you look at the cells and NK cells that express particular gene sets, particularly TNF alpha response, interferon gamma response, you see that there's a high expression of those genes. There on the left, you can see exactly what genes are underlying these differences too. Here's the SNP in this locality. It's a SNP that's near a gene called KLRC1, which is an important gene for NK regulation. We have some ideas about what we think is going on. HLA-E interacts with NKG2A, which is the protein that is encoded by this gene, and that controls NK activation. So we think the SNP is interacting with the gene that's important in NK activation, and that's why we see an altered response to TNF or interferon gamma cytokines. Turns out that the SNP is associated with psoriasis risk. So this is very interesting, not just from a biological point of view, but actually has clinical implications. We feel like we're starting to find things with this result. The data sets aren't really large enough. We know from flow cytometry studies where people found SNP associations with different cell states that the data sets had to get really large before we were able to find large numbers of loci. So we hope to get these data sets in the future. As you can see, this is a power analysis of how much variance explained we think a typical SNP might have, and how much power gains we have over as our sample set gets larger. So this method is published. It's in Nature Genetics earlier this year. We are able to actually do this work with any single cell profiling modality, any tissue. New single cell data sets, data integration will hopefully increase our power and sample size. And GeNA may reveal a broader catalog of csaQTLs across human tissues. And so we think the important thing is obviously getting more of these sort of genotype data sets from more populations, from more diseases, and we think this method will be useful. So to summarize, before I stop, our group is very focused on developing methods to define human disease mechanisms. Autoimmune diseases are a key focus for us. Single-cell genomics, we think, is a powerful tool. Defining key cell states associated with disease or disease subphenotypes is something that's important, that we're using single-cell genomics. It requires critical methods. We talked about neighborhood methods. We talked about GeNA and cNA, which have been using us to find case control differences and genotype differences. Both of these strategies exploit cell-cell similarity and also sample-to-sample variation. And that's been really important for us. And it sort of trades off the benefits of high granularity versus observation of shared trends across samples. So future direction, we're excited about larger, richer data sets, and we're excited to use our methods on them. We're also thinking a lot about spatial genomic data as they become available, and we're starting to think about how these same frameworks can be used in the context of spatial data. And we're also developing experimental strategies to confirm the effect of SNPs on cell states by doing gene editing with CRISPR. So with that, I'll just thank the lab. Here's a bunch of members of our lab on our annual lab retreat up in northern Massachusetts in a town called Salem. I thank you for your attention. And with that, I can stop and take some questions.
Thank you, Professor Roy Chaudhry, for such a wonderful, detailed talk, and a highly technical one. I think for me, from my end, I'll have around 2 questions. And really, single cell analysis is, I think it is quite an exploding field, but I think for me personally, I came across it. I think like there is a paper that came out, I think like 2 years ago, about single cell analysis with the graph neural networks. And that's how it caught my attention. But the question like I have would be about, we are talking about the rheumatoid arthritis, like all these cells that we are talking about are within, sort of to say, at within patients of rheumatoid arthritis, and there is nothing else involved.
Yeah, great question. So this was probably a little bit confusing. So I presented work on a few different data sets. So one of the main data sets we've worked on is data from joint tissue, inflamed tissue from patients with rheumatoid arthritis. So the examples I gave where we said we're trying to look at differences between different patients, that's largely from that data set. a lot of the data set, the public data, such as the 1K data where we did a lot of the genetics, that's a large data set, and that's mostly from blood. So some of the largest single cell data sets are blood data sets because, blood is easy to obtain from patients. It doesn't require a biopsy. It doesn't require tissue disaggregation. So when we've thus far looked at the effects of genetics on cell states, it's been done mostly in blood samples, not so much in sort of tissue samples. And those individuals are not people with rheumatoid arthritis. Part of our thinking is that we expect that the effect of a SNP that puts you at risk for a disease, that there should be a molecular effect of that SNP on healthy individuals too, because that SNP puts healthy individuals at risk of disease.
Thank you, Prof. Another thing is about we are looking at single cells, and for this context of rheumatoid arthritis, is there anything we can say about the cells sort of like at the boundary between like the rheumatoid arthritis and the cells that I would say within the health region? I don't know whether it makes sense.
Yes. Yeah. No, it makes perfect sense. So at the most coarse level, we know in rheumatoid arthritis that you have many more immune cells in your synovium. So you have more T cells, more B cells. Um, but in a way, to me, the more important question is not just do you have more immune cells, but is the distribution of the immune cells different, right? Because we think these, for example, T cells are incredibly important to rheumatoid arthritis pathogenesis. And we think that there's a certain type of T cells that reside in the tissue in health, but they become different when you have disease. So Deepak Rao, one of my colleagues, described in great detail T peripheral helper cells with my other colleague, Michael Brenner, working together and also with me. And what we found is that these T cells are not only are T cells abundant in the blood, but T peripheral helper cells are disproportionately expanded compared to all T cells in rheumatoid arthritis samples. So there are differences between health and disease. Of course, there has to be. And to me, the most important question is not just what are those differences in gross terms, but what are they in fine terms? And then finally, that brings me to the third question, is which of these differences are reactive? That is, they're kind of happening because there's some kind of process going on. And which of them are actually driving the disease? Which of them are the ones that are actually problematic? Which of them are the ones that if you intervene on, things will get better? And I think that's where the genetics is really useful. Genetics points hopefully to the cell types that are likely to be causal.
Thank you, Prof. Lastly, for me is when you look at GeNA, it is sort of like it is scanning the entire sort of distribution. And from a computing perspective and a computing mind, how can we reason about this algorithm within GeNA itself? Like, what can be said about it?
Yeah, so the GeNA method is really interesting. So we scan the whole genome, right? And every time we test a SNP in the genome, we're testing the whole distribution of what's happening with cell states. So it's a method that has to be very fast, right? Because
Yes,
we
yes,
are
yes.
doing so much testing. It's also a method that has to be very, very statistically accurate. So GeNA has both of those properties. Fast, because you're testing so many things. GeNA does a lot of work. The method has a lot of work under it to make it a linear method, which is essential. That's part of why we aren't just crawling around the nearest neighbor graph and testing every SNP by doing some complex nearest neighbor graph thing. Rather, we're converting that nearest neighbor graph into neighborhoods and neighborhoods into neighborhood abundance matrix PCs, and then we're testing at that level. Second thing that's really important about GeNA is its statistics. Any of these tests where you're testing a million SNPs, millions of SNPs, if your statistics are inflated at all, you're in danger of finding false positives that are uninformative. So again, we spend a lot of time to make sure that our analytical p-values for GeNA are very accurate.
Wow. I think that's all I had from my end. Just the algorithm for me was fascinating. And I don't know whether the members on the call do have questions. The university here has closed and people have traveled up country, going back home. And yeah, it explains the attendance, Prof. Yeah. So
No worries
I
at all.
think
Thank you so much.
yes, yes.
Sorry.
All right. I don't think other members have questions. I would like to thank Professor Richardly. Really, thank you very much for this opportunity and for allowing and agreeing to teach us about this wonderful work. We really appreciate it. Getting time off your busy schedule and talk to us.
Thank you so much for having me, and look forward to speaking to you again soon.
Thank you very much. So to the rest of the members, those that are still here, bye-bye and thanks for attending, and goodbye to Professor Richard Lee.